Good afternoon, esteemed speakers, ladies and gentlemen. A very, very warm welcome to this fourth webinar in the Brain Matters series, hosted and organised by the Human Brain Project. My name is Katrina Sickle. I'm a broadcaster and moderator, and I have the privilege of bringing this 75-minute discussion to life, a discussion on brain simulation science and technology. Now, our aim, if you've been following us to date, and some of you may well have been, with this whole series is really to present scientific brain research, some results, some methods from within the Human Brain Project to an audience that is very broad, ranges from the molecular biologist at one end to the curious non-specialist at the other. Now, whoever you are, no matter your area of expertise, you are invited to write a question in the chat bar of your social media platform at any time during the course of the event, and I will be able to pose some of those questions to our three fantastic expert speakers towards the latter end of that 75 minutes. Of course, the rules are keep your contribution short and to the point. That will really help, and our team, our HBP team, will be curating those for brevity and relevance. You can also, of course, share words of wisdom on Twitter at any time. It's #BrainMatters or @HumanBrainProj. In today's edition, as I said, I'm joined by three eminent scientists. I'd like to ask them to join me now on screen, please, so you can see who they are all together before they individually present. Well, thank you. I love the different styles of waving there. Michele, you were quite sort of regal, and Jeanette was very excited, and Eduardo was a little bit like this. We'll see how that correlates with your presentation. So, Jeanette, let me start with you. You can see her name there, Jeanette Hellgren Kotaleski. She is Professor in Neuroinformatics at the Department of Computational Science and Technology at the School of Electrical Engineering and Computer Science, so that's KTH Sweden. And the main focus, I mean, this is a very short bio, the main focus of her research is to use computational modelling to understand the neural mechanisms underlying information processing and learning in motor systems. So thank you very much. I'm sure you could say pages about yourself, but those are the top lines. There we also have with us Eduardo Ros. He is full professor and head of the Computational Neuroscience Lab at the Department of Computer Architecture and Technology at the University of Granada. And a few of his particular research interests are spiking neural networks, high-performance real-time computing, and its applications to robotics. So you're going to see that in his presentation today. And of course, here we have a gentleman, Michele Migliore, and he is head of the Computational Neuroscience Laboratory at the Institute of Biophysics of the Italian National Research Council. And his previous work with the Human Brain Project has focused on modelling single cells and the hippocampus. And again, you're going to hear from his area of specialism today. So, ladies and gentlemen, each of these three good people are going to present some of the top lines of their work and how it all correlates together within the project. They're going to present individually, but before they do, I'm going to need to frame these discussions from you. So I'm going to ask Michele to stay with me, and I'm going to ask you, Eduardo, and also Jeanette, if you can kindly take a breather backstage, and I'll have a quick chat with our first expert speaker here. So welcome. You have a little, it looks like it's snowing inside where you are.
Yeah, there must be some glitch on with the camera, but
that's all right. It looks really quite festive. Let us get straight to the heart of the matter.
Yeah.
What is brain simulation science?
Well, it is a powerful way for scientists to implement a virtual model of neurons and network. This is important because it can help with interpretation of experimental findings and to eventually lead to a better understanding of how the brain works. For example, it can help understand how changes in the brain structure can affect neuron communication, and this can lead to figure out how it can alter behavior and cognitive processes. This is one of the example.
Okay, so to give it to amplify then, to make it very concrete, could you give us an example of a couple of sectors perhaps where simulation is used?
Yes, well, actually, brain simulation is one of the fields in which simulation plays a paramount role in helping real-world life and applications. For example, pharmaceutical companies are now using molecular modeling to guide the design of new drugs. Or, for example, in aeronautics, engineers heavily rely on modeling. No airplane prototype goes into real flight without passing the simulation test first. And also, of course, business and finance modeling is used in the economic world to forecast revenue, growth, market changes, and things like that.
So those examples there lead me naturally to my next question, because we've looked at what it is and some examples of where broadly it can be used. But I think a really important question is, why do we need it?
Well, the brain, again, is it's a complex system that can benefit from modeling and simulation science. Let me explain with one example that it is very common nowadays. If you want to understand how the brain works, we need to find a strong theory for it. For example, if you want to go to Mars, then observing the relative location between Earth and Mars will not give you enough information to land the space shuttle, because we need a theoretical model to calculate the trajectory to go there. Okay, in this case, the law of gravitation. Analogously, experimental data on brain activity alone cannot give us enough information to understand how the brain works. We need to find one or more laws of brain activity, and this can be done more easily with the brain simulation science.
So what can we learn from it then that couldn't necessarily be obtained in another way?
Well, with the brain simulation science, we can do what we call virtual experiments that, for technical or ethical reasons, cannot be done in vivo or in the laboratory. And once we have a model, we can ask questions and get answers that can be directly related or tested or applied to real-life problems. As I already mentioned before, it can be used for predicting the dynamics of the network communication. But today, we are going to focus on the processes related to the three brain regions, like the cerebellum, hippocampus, and the basal ganglia.
I asked you at the outset just to make it very concrete and graspable, and I said, give us some top-line examples where simulation is used. So are there any other sort of applications that you concretely, particularly in the field perhaps where you and your colleagues are working, can help explain its benefits?
Yes, there are many fields in which the benefits from brain simulation can be applied. For example, in the pharma, the clinical field, the robotics, and more generally in the industrial commercial fields. Brain simulation science can greatly speed up the search for new drugs, for example, to treat mental disorder and diseases, because the data from these kind of brains can be used to create models to understand how pathologies alter the normal structure and function of the brain under these conditions. Other applications may include helping improve quality and efficiency of robotic application, for example, or brain-machine interfaces, and help to improve the interpretation of clinical imaging, or helping discover new products for industrial commercial application. I will discuss a few examples of this in my presentation.
Okay, that's really helpful. I think we really need this broad picture, and it's interesting, just to reinforce what you said, Katrina. Look, experimental data is not enough. And for various reasons, there are certain experiments that we just can't do in the laboratory, and it's very, very critical. We've heard about some of the areas and the sectors, and we'll hear more in the presentations that are coming. My last question would be, sounds fascinating, important, has a very clear place. Do you think that there's perhaps a risk that it could be seen as the solution to every brain problem?
Well, yes, there is this risk, but we need to be careful and understand that the brain simulation science field is complex, and it should not be seen as the solution to every brain question. It is one tool that could be used to answer a set of questions that will be increasing in the future, but not that tool. We are still in a rather experimental stage, and a lot of things needs to be learned and done. However, they are very promising. There are very promising results around, and they can pave the way to more discoveries and potential breakthroughs in brain science.
Okay. Thank you very much. You're not released yet. Actually, your work here is not done because you happen to be the first of our three experts who are going to present. And just tell me briefly, for the benefit of our audience, what are you going to focus on in your presentation today?
Well, I'm going to focus on the main field of my research, which is the hippocampus region, which is involved in many brain diseases, and it is also involved in higher brain functions like learning and memory, spatial navigation.
Fantastic. Thank you very much. I'm going to stay on screen so that I can watch that presentation and listen to you. I'm going to let you get set up there, so just so that we can make sure that when you share your presentation, we can all see it. So I will give you a moment to do that.
Done.
Perfect. Well, I can see it, so I can leave the floor to you.
Okay. Good afternoon, everybody. So let's start by assuming that you want to model a brain region. There are many ways to do this. One very popular way, for example, is to treat the brain region like a black box, and then figure out a function or an artificial network or an algorithm or whatever we are smart enough to implement in such a way that after some training, of course, the function or the black box is able to give us the correct output to an enormous amount of, or in general, an arbitrary input. This would solve the problem of having a brain region modeled in such a way that we don't care, we are not perfectly aware of how the black box works, but we are just interested in getting the right output for any input. What we do in our work with the brain simulation science is to open the box. We call it a bottom-up data-driven approach. In this approach, we open the box, identify the components, and figure out how they work individually, how they are connected, and how the network evolves, and finally implement a model of all this. At that point, I will show you later an example. We are ready to do in silico experiments which can be related directly to real life. So let's start from the basic components, which in this case are the individual neurons. There are excitatory or inhibitory neurons in the brain. So for example, for the hippocampus, we have been collaborating with groups of experimentalists at UCL or in Budapest. They gave us morphologies of the pyramidal neurons, for example, of the interneurons. These are actual 3D reconstructions of neurons. They gave us also the experimental recordings. These are traces from the different types of neuron. And at that point, using a model that we have based on the so-called Hodgkin-Huxley equations, we implemented the model for each type of neuron. And these are a couple of examples. Here you see the original experimental traces. Here you see the computational model. And this allows us to have a very tight connection with experiments, because we can make a direct comparison between our modeling results with in vitro or in vivo recordings. This makes easier the interpretation of experimental findings in case there is some puzzling experiments. And we can also do, of course, experimentally testable predictions. Let me give you an example of the application of the model. Here we have a model of Alzheimer at the single cell. In this case, we have a model of pyramidal cells in the hippocampus. We activate a bunch of synapses in the dendrites, and we implemented Alzheimer progression by assuming that each branch, each dendrite of the neuron is affected by Alzheimer according to the experimental data that showed us how the different ion properties can change when they are affected by Alzheimer. And when we do the model, we can see the actual effect of Alzheimer. And not only that, in this simulation, you are going to see here some moving stuff. And let me explain you. So we are activating the synapses. the synapses will generate some background activity, and eventually they will generate a spike and action potential of the soma. This is the signal that is transmitted to the rest of the network. Under normal conditions, you see several spikes because the synaptic input is strong enough. However, if I simulate Alzheimer by applying all the experimental data telling us how the Alzheimer changed the properties of the membrane, you see that the neuron doesn't work anymore, at least it doesn't work anymore as it was in the control conditions. So at this point, what we can do is figure out or predicting what we can change to make the neuron work normal again, even if it is affected by Alzheimer. And when we do this, you see that the neuron goes back to normal state. And this could help the pharmaceutical companies, for example, to figure out how to target a molecule that is able to implement what our model is predicting to happen to treat Alzheimer, for example. And we did this for many other different cases. You see here a few papers in the past that have been published, not only for Alzheimer, but also for epilepsy and other issues. Once we have the single cell model, we can go and figure out how they are connected. In this example, for example, you see the pyramidal cells, the different population of interneurons that are modulating the output of the hippocampus. And so we, again, starting from experiments, we implemented a model for all kinds of synaptic connections. And at that point, we were ready to implement a model of the hippocampus in the rat, of course, because we don't have yet data from the humans. But at this point, we have a full model of the rat hippocampus, and we can do in silico experiments and be directly related, directly in connection with the experimental data at the level of the in vitro experiment, for example, when the experimentalists do the experiments on the slides, or for example, imaging, because we can calculate the local field potentials, the equivalent of the electroencephalogram data for the hippocampus and so on. So the idea is that once we have this system, we can now reduce it to make it more simpler and run in real time and connect it to a robot in such a way that we can have a robot with Alzheimer, a robot with epilepsy, and we can figure out what we need to change in the circuit in such a way to treat and make it an epileptic brain or an Alzheimer brain works in a better way. So from here, what we can do is to also figure out if there are already in the hippocampus some specific cells or circuits that are connected to cognitive functions and try to make a real-world application. So for example, let me give you a couple of examples. This is a recording from an experiment in which a rat is implanted with a bunch of electrodes, recording from individual pyramidal cells in the hippocampus. And you see these little colored dots represent firing the signal emitted by different neurons in the same region, the CA1 regions of the hippocampus. And you see that they fire an action potential every time that rat is in a specific spatial location within this room, okay? We call it place cells. And those are in vivo recordings, of course, because you see the rat going around. But let me give you another example. This is an in vivo recording from a human, again, from the hippocampus. And when they show the person different pictures, you see the noise. This neuron, for example, was selected for Marilyn Monroe. So it means that every time Marilyn Monroe, not only the image itself, but also the name was appearing on the screen, that neuron signaling that object, in this case, a picture of Marilyn Monroe. Of course, there are different neurons tuned for, at that point in their life for different object or people. In this case, this other neuron is selected for this guy, Josh Brolin. I have no idea of who he is, but I mean, the neuron is recognized him and not Marilyn Monroe. So this gives you an idea that there are already in the hippocampus some cells type that have specific functions. So this gave us the idea to implement a network in which we put place cells, object cells, persistent firing cells, which is another population of neurons that are saved in the hippocampus. And then the network was connected with the plastic synapses, which means synapses that change with time and activity. And we connect the network to a robot, equipped only with a camera. And when we let the network evolve, we see this. The robot goes around, it will bump against the wall because it has no control. So of course, it is a very proof of principle implementation. And when the network showed us that it needs only one pass to remember this correct sequence of monitors and go directly to the exit, and even avoiding walls or obstacles. A single pass of training trial, no matter how long is the sequence, would suffice. This is exactly what happens to us as a human when we go around and visit a museum, for example, and then we stop and say, okay, I would like to go again and look at a specific piece of art. We go back and we don't need a lot of training. We do it just after one time. This is a more complex example with a maze, a T-maze. These are the evolution of the weights connected to each object. And you see that as the robot finds that this is a wrong exit because it's looking for the green spot, it goes back, he forgets the synapses, he forgets the objects, and then the next time he knows that this is a wrong branch, takes the other one and goes to the exit. And this again, in a single learning trial, the robot is going to know exactly where to go in the recall phase. So at this point, of course, we are going to have the hippocampus black box ready, but equipped with all the circuits that we know it is they are in the hippocampus, and we can connect this region with any other region that are going to interact with the hippocampus in the real brain. And Eduardo is going to talk about the cerebellum and the work that he has done with based on the model of Egidio D'Angelo. And then we can, of course, wait for the model of the cortex and other brain regions. And then eventually we can reach a point in which we have the full brain modeled, and then we can begin to make real prediction, experimentally testable prediction on cognitive functions. With this, I thank you. This is the large group of people working on the hippocampus within the Human Brain Project, but also outside, for example, for the Blue Brain Project, UCL, my group here at the CNR, and also in Budapest. And I think, of course, this is part of my lab that has contributed to this work. Thank you.
Thank you very, very much. Thank you. And I'm glad you showed them because, goodness, it's absolutely fascinating. I love the way you took us step by step through things there, starting with the black box, then the single cells coming to the connected cells and the robot, and having a look at those examples there in the learning trials, and then coming finally to the links and the connections with other brain regions, which is also part of features, obviously, today. Just one very quick question before I move on to your colleague, Eduardo. You said there, what we can look at when you were talking about Alzheimer's, but, other diseases, what we can look at, what we can change in a neuron to see, how it's affected, how it could be made to work again. And you said this can help pharma. They could target a particular molecule, say, for treatment. It can really kind of give them a steer in the dark. So when we're talking about that, what sort of time frame are we talking about when you say?
Well, I mean, for specific diseases, I think that the time frame for which we can give a kind of suggestion to the pharma companies is a few months, because we already have the model ready for the single cells. So we are ready to test several different combinations of modification of ion channels that could really help the search for new drugs in a few months. Then, of course, this is basic science. The pharma has to go through all the usual process of marketing a drug, and this could take years. But again, they are going to say they may save billions of dollars in basic science research.
Although I think the expectations of things taking years, now that this the vaccine for COVID has come, that's going to shift the demands on the speed of things. And by the way, before you go, on a lighter note, Josh Brolin is an actor. There we go.
Okay, good to know.
I'm letting you know. Anyway, you very kindly, right at the end of your presentation, talked about, you know, the connections with other brain regions. And of course, we're going to hear exactly about that, the cerebellum, from our next speaker. So, Michele, you get a chance to just relax off screen and can follow bye-bye the presentations of your colleagues. So fantastic. We've heard that the focus of Michele's work is on one particular brain area, the hippocampus. So we're going to find out a little bit more from our next speaker. So if I can ask you, please, Eduardo, to join me, that would be fabulous.
Thanks.
Hello.
Hi.
It's quite nice to be the second one. You don't have to break the ice and you don't have to wait till last. You're sandwiched in the middle. So sandwiched in the middle as you are. Anything else you want to tell us, or is it all a surprise apart from the fact that the focus, the brain region focus, is the cerebellum? Anything else you want to say, how it complements the work of your colleague?
Yeah, well, it's a different brain area. It has also other ingredients as we interface with real robots, so it's a bit different, but it shows another kind of applications we can address with these kind of simulations.
Okay, thank you. So let me allow you to get ready to share your screen, and then I can tell you if we can all see it. Yes, that's great. I'm going to be quiet now, and I'm going to let you present. Thank you.
Okay. Yeah, I will talk about neural simulations and neural robotics. So how we integrate neural system models with robots performing specific tasks such as coordinated and accurate movements. So I will specifically talk about the cerebellum and how we address a scientific question of how the brain or the cerebellum acquires skills from interacting with the environment, and how we can use that for real-world applications, such as accurate control of collaborative robots that have elastic components of their joints, such as muscles, which make them inherently safe on one hand, but make them also challenging to be controlled. When we do this kind of simulations, we have different approaches for different purposes. So as Michele indicated, data-driven simulations for integrating realistic experimental data into models. I will focus more on other two approaches, hypothesis-driven models, in which we explore how neural microcircuitry computational capability is based or can be based on neural properties or the neural dynamics. And I will also show how we can use functional models embedded within a closed-loop brain environment experimental setups to explore how a task or a performance is based on computing principles of the neural substrate. So here, for this kind of simulations, we need different ingredients. And the first ingredient is the connectome, the neural microcircuitry, or the wiring diagram. We see on the left picture a diagram by Ramon y Cajal more than 100 years ago, in which already outlined different kinds of neurons, how they are connected, related within a network. We can abstract this model in the middle picture, in which we also indicate different kinds of neurons, different connections, how the information flows, and more details about it. And we can further simplify it to this synthetic network on the right-hand side, on the right picture, where we have a well-abstract network with all the connections well-defined and different neural populations dedicated to specific joints of a robot when we integrate that with a robot, as I will explain later. So another ingredient is the neural plasticity mechanism, the learning rules. So the brain adapts and learns by changing the different synaptic connections that can get stronger or weaker depending on the stimulus and the produced responses. So here is just a very simple example of how the information flow goes through a simplified cerebellar structure, with the information going through the mossy fibers connecting these granular cells whose axons form the parallel fibers that connect to the Purkinje cells and then to the DCNs. With this simple structure, we can also study how different adaptation mechanisms take place at different sites of the network. This adaptation mechanism will follow certain equations, and we can check how these help and complement each other for a specific task. Then we have also different cell types in a network. And when we do this kind of simulations, we can do that at very low level of detail, at a molecular level, or with simplified models as here, in which the behavior of a cell, the dynamics, are described by a few differential equations, as the one shown here, with a few parameters that we can tune to fit the dynamics of a specific behavior. So here we see the black dots are experimental data from Egidio D'Angelo, and we tune these parameters to fit the dynamics to match these experimental data, so that we arrive at a granular cell model, so to say. With this kind of simplified neurons, we can simulate thousands or millions of neurons and then integrate them with real robots, as I will explain. Then finally, we have also to define how we embed this neural structure within the whole neural system. And in this sense, the cerebellum is in a privileged site, receiving motor commands. These motor commands are also delivered through the brainstem to the spinal cord, and then to the final actuators, the muscles. The cerebellum will also receive early sensory motor feedback, so that it can match the motor commands, the sensory motor feedback, and produce corrective terms or predictions of the sensory consequences of the actions, so that different adaptation mechanisms can be implemented to perform these accurate movements. And this is just an example of a control loop and how we implement that in a control scheme. And all this we test within experimental setup. This is vestibular ocular reflex, the VOR experiment, which is widely used to study how the cerebellum works. Here, the goal is to fixate the gaze despite the head movement. So in this case, the subject, the patient, or the animal, it learns to perform eye movements to compensate for the head movements. When we also do this with animals that are placed on a rotating platform, and when the platform rotates, the animal learns to perform these eye movements to compensate for the head movements and being able to fixate the gaze. Here we do these kind of simulations with simulated robot. On the top right, we see the learning curve. So the error becomes lower and lower, so it is becoming better at the task with different iterations. We see on the left that before learning, the head is moving and the eyes are also moving, but they are not compensating for the head movement. And this is why below we see that the scene is not static. While on the right, we see that after learning, the head is moving and the eyes are moving, compensating for the head movement. And this is why at the bottom, we see that the scene stays static. And this is what we do when we walk around, when we jump, when we move around, and we are able to fixate the gaze despite all these moving components in the head. And this is a very similar simulation, but in this case with real robots. So we are doing the same before learning, after learning, but we are addressing real-time challenges. So the simulations need to go really, really fast, really well in real time. We see again that at the bottom, after learning, the scene stays static, so it has performed well. And then I will explain briefly another example of how we use all this for another task, a different task, which is in this case, arm movements. We see the same ingredients I have mentioned before. On the left top table, we see different cell types. We will use in this simulation more than 60,000 cells, more than 30 millions of adaptive synapses. At the below in the other table, we see that different cell types, granular cells, Purkinje cells, DCN cells, are defined with different parameters that help us to describe how they will behave, and how these components are integrated on the right picture within a neural structure with the different connections and adaptive synapses. And then how all this is integrated within a control loop at the bottom. With this kind of setups, we can do this kind of simulations. One moment. Here we have the robot, which is performing a circular movement. So after learning, it does quite well. Here on the left picture, we see the error curve. So, and we see with the dashed line what we would achieve with an industrial robot approach. And on the right top plot, we see the activity of the 60,000 neurons, the spikes that they produce. And at the bottom, we see the torques produced by the cerebellar model. So in the simulation, the robot is doing this circular movement. Now we attach a tool to the robot so that it has to learn the new dynamics. It's the same as when we use a hammer or a racket and we learn to use it, and it becomes an extension of our arm, and we can do accurate movements with them. So we abstract the model and we are able to move it correctly, to manipulate it correctly. And this is something that humans are particularly skillful for doing these kind of things. When we take it out, it takes some iterations and then it comes back to the basic model. It doesn't need to learn again, but it shifts to the basic model. We can address a more challenging task, in this case with an elastic force. We will attach an elastic component to the arm so that the robot will need to do this circular movement within a force field. So we see here that is now it's quite a challenging task. It will learn, so after some iterations, the cerebellar model will learn this force field, and it's able to do this circular movement in the presence of this force field. And this is done with a compliant control scheme, very smooth and safe for the operator. We will see now how an operator can interact with the robot, and with a single finger you can push the robot and move smoothly away from the task that it's performing. And you see here that the operator comes in and with a single finger can push the robot away. And this is something very different to what you would see if we were using an industrial robot with rigid movements and very, very high torques being applied to the motors, which would be also potentially dangerous for the operator in case of malfunctioning. You can also step or bump into the robot and it would be quite safe. So in that sense, it's a compliant. So here we have the operator manipulating the robot. So in that sense, it's safe and compliant, well, it doesn't represent a threat to the operator. And finally here, the last example is how you can bump into the robot and well, it will move smoothly away, so not representing a threat. So this kind of, I just have shown different examples. As Michele said, you can do these data-driven models to integrate what we know from experimental data. In our models, we also fit the cell types to represent some of the properties of these different cells. But even with these simplified models that we have used, we can explore how the system works. And we can also use this for real-world applications, as in this case, accurate control of collaborative robots, and learn about ideas from biology, how it addresses biologically relevant tasks, such as accurate and coordinated movements. And this is all. So this is the team, and well, thanks to the team and to all these international collaborators, as Egidio D'Angelo, Angelo, Arleo, and others.
Thank you very much. Thank you. And I think it's interesting there where you also talk about international collaborators, because I think that is also part of this project, and really having this very, very broad reach and encouraging that kind of multidisciplinary and really international approach. And that also echoes, I think, what Michele spoke about at the beginning, some of the research that had come to his work in the project from the outside. What I absolutely fascinating, and I asked there just a moment of your time before I give you a little bit of a breather before I ask our third speaker. I spent a bit of time at the beginning with Michele saying, well, give us some real world, some sectors, or some real world applications. You did speak about that. Is there anything that's particularly important to you as an application? You know, you said there where it can be used. What's very close to your heart? Where do you think, you know, it really is a big societal benefit, this simulation science and its path forwards?
Well, we can learn about how the brain works. So in that sense, there are a lot of biomedical applications, as Michele also mentioned, in the pharma industry. I'm also focusing on how we can use that for real world, well, in this case, robotic applications. And this, well, I have shown an example of when you manipulate this kind of robots, they have these elastic components that make them quite challenging. So in that sense, industrial control setups will not do quite well. So the accuracy you get with that is not very good. So you have to learn about how the brain does with this kind of actuators that we have. So we can also take benefit of that. Mm-hmm.
Absolutely fascinating. There are lots of questions coming in already for our first speaker, and doubtless there will be plenty for you. So just take a brief break backstage, if you will, and I will shortly ask our third speaker to join us. So absolutely fascinating. We're going to speak more. I'm going to ask all three of our speakers just before I come to audience questions, you know, what is it about all of these different brain regions cooperating, collaborating, depending upon each other? And we're going to hear about another region now from our last speaker. Can I please ask Jeanette Hellgren Kotaleski to come back? Thank you. Thank you so much for your patience. Absolutely fantastic to have you join us, because it's such an important piece of the puzzle, because we've heard from the experts, world experts, in the hippocampus and cerebellum. And at the beginning, when I was speaking with Michele, he said, you know, simulation science can be used to monitor the dynamics of neuron network communications within specific brain regions, but also the collaboration between them. So yours focuses on another region. What are you going to share with us in your presentation today?
Yeah, so I'm going to exemplify how modeling and simulation have helped us to understand different aspects of this third brain structure called the basal ganglia.
Okay, thank you very much. Let me give you a moment, as I did the others, to just set yourself up with your presentation and reassure you that I can see it. So if I can see it, everyone can see it. There we go, perfect. The floor is yours, Jeanette.
Yes.
Thank you.
So as said, I will sort of illustrate what we did in the basal ganglia to understand it better using models. But first I will take a step back and say some words about what you have already heard, to put these different modeling philosophies in some context. I will just start with this obvious challenge that to understand the brain, this is a challenge because we have to bridge between molecules, cells, networks, network of networks, and then various types of behaviors, cognition, perceptions, actions, and so on. And the challenge is, as was said, that at each of these levels, we have many dynamic and interactive processes, and also there are feedback loops between these levels. So obviously, this is why we need simulations and modeling in addition to data and data analysis. But when we build models, I mean, the biological scale is, of course, a very important constraint. What level do we want to model at? But this is not the only thing that decides how we build the model. We can build the model in a very data-driven way, as you have heard. You put pieces together, pieces of data together at a certain level, and for instance, see how things emerge at the next level. Or you might already have a very specific hypothesis to test. For instance, I believe that this cell type can lead to a particular phenomenon that you can measure in experiments, and maybe I build a minimal model just using those few components. At the same time and at the same level, you can also build models in a very so-called mechanistic manner. That means that model variables and parameters are supposed to map to biological entities, but you can also simplify the description. And we have sort of heard all of these kind of models exemplified. And eventually, to understand the brain, we have to fill all this three-dimensional space. And the purpose of the model, that sort of decide where in this space you should be. Do you want to understand the real brain and even target things like ion channels, receptors, to do drug discovery? Then, of course, you have to represent those in the model, as you have heard. But if we want to extract principles for AI applications, or for how the brain learns, or if we want to create this sort of brain theory, more generic brain theory, then it's also useful to simplify and compare what you lose, and use hypothesis-driven modeling. Just wanted to mention that I think that since different labs work in different parts of this sort of modeling universe, it's extremely important with the reproducibility of actually the whole modeling process, ideally, and also that all the models are interoperable, so we can plug them together and reuse them. And fortunately, this is something that is supported by eBrains and HPP. Here I have mapped on this scheme what you already heard. So Michele highlighted and exemplified how you can build cellular level microcircuit models in a quite standardized manner, and this has also been done for the cerebellum. And then both Eduardo and Michele, they discussed functional models, how you can then simplify the models and to test some hypotheses, how the hippocampus and the cerebellum in this case could support some particular functions. And now I will do the same for the basal ganglia, but then some facts first. So the basal ganglia is a subcortical structure. The input stage is called the stratum, that is the biggest one. And this stratum receives convergent input from the whole cortex and part of the thalamus. And this information is then conveyed through different pathways through the basal ganglia. It controls motor centers in the brainstem, but there is also information going back via thalamus to cortex and actually back to also to stratum. Neuromodulation is critical for this system, and indeed one of the basal ganglia nuclei is in itself producing dopamine and signaling with dopamine. Also, interestingly enough, the basal ganglia is very much evolutionary conserved, and it has sort of this very similar organization when it comes to cell types, neurotransmitters, all these different nuclei, and how different cell types are connected within the same nuclei, but also between the nuclei. This is really evolutionary conserved from one of the oldest living vertebrates, the lamprey, to man. And that also suggests that the basal ganglia has kept some kind of very generic function throughout evolution, at least I think so. So what is a function? Well, as you see here, the basal ganglia is sort of a bridge between, in this case, cortical activity patterns and thalamus, and the behavioral repertoire that you have. And so it's, and as you know, I mean, the behavioral repertoire is quite limited. You can't drink coffee and talk at the same time, for instance. So you have to choose wisely and prioritize what you're going to do. And this has been very neatly sort of described by Arber and Costa recently. They wrote that if the cortex broadcasts a wish, the basal ganglia will decide whether the wish should be fulfilled or not. So it's involved in sort of action selection and decision making, what to do in a particular context. And this is all something that has to be refined all the time and adjusted based on how the environment changed. So here, reward learning is important to adjust these decisions. Obviously, since it's a link between cortex and thalamus and the behavioral repertoire, if something goes wrong, you get motor disturbances or neuropsychiatric disorders. And maybe the most well-known is Parkinson disease, of course. So here I have now tried to map the different type of basal ganglia models onto this possible universe of different type of models, and probably I missed several, but this is sort of proof of concept. Here I have also indicated something I called subcellular model, where you can study plasticity and learning. And I should just say that this has also been done for hippocampus and for cerebellum, but it wasn't highlighted that much today, even though they used simplified models in their of plasticity in their functional models. But I will come back to that and try to exemplify. Also, I wanted to point out what Michele also said, that within HPP, I think an important contribution within HPP is that for quite data-driven mechanistic models at this kind of scales, we have developed standardized workflows, which means that different labs, if we share the data and the software tools, in principle, different labs could reproduce the same type of model. And this is extremely useful, of course. Okay, so one example of a microcircuit model, a quite mechanistic data-driven model of a piece of the basal ganglia. This is the stratum, the input stage, the biggest part. Have also been built for the basal ganglia, as was already illustrated in the campus. And then we need these pieces of data that was already mentioned. So we need to know the macro anatomy of the stratum, of course. We need to know cell densities, which cell types are there, how are they connected, the electrophysiological properties of the different cell types, including neuromodulation in this system, properties of different synapse types in the system. We need to have reconstructed neurons of the different types. And then we can also use this structural information to try to predict how the neurons are connected, where are the synapses, so we can try to predict the whole network. And here, these automatized modeling pipelines and software tools are of course crucial. And then we can come up with a simulation of a part of the stratum with different cell types. This is an illustration of that. And here we give sort of a cortical command. Not surprisingly, the activity goes up. And here we give instead neuromodulation via dopamine. And then we see other changes and so on. And as said, this can then be used as a test bed where we can try out things that are difficult to do in experiments, for instance. And you might know that there is something called HPP Live Papers, and if you want to see more of the data that went into this in the process, you can go there and check it out. So that, and now we come to an example of a so-called functional model. And I should say that when it comes to the basal ganglia, which is involved in sort of action selection and so on, there has been a lot of functional models built in various labs. But here I wanted to show an example how one could pre-train a basal ganglia module or pre-configure a basal ganglia module to perform sort of goal-directed behavior based on the control of this motor infrastructure or this behavioral repertoire that you have. And then we have taken the example of the lamprey, which we have studied in Stockholm for many decades. So we have a good understanding of how the spinal cord can generate swimming movements. We have detailed models of that since a long time ago. And we have a good grasp on how the midbrain and brainstem in turn control these spinal cord networks. And then we can plug in a simplified basal ganglia sort of module to try out how can output from that control different populations of neurons in the midbrain and brainstem to control motor behavior that should mimic the real thing. And this is just sort of a toy example of a lamprey that wants to swim again towards cones, but it has learned to avoid the red one and hit the blue one because then it gets rewarded. But this is sort of a simulation where inputs from a basal ganglia structure can control turning, starting, stopping, and this hitting of this blue cone. So I guess you get the point how to use models in this case. This is a little bit, this is also an example where you can plug together different models. And I mean, if we had this detailed knowledge of a mice or a monkey or whatever, then we could redo this simulation. But now we only have this sort of detailed models at this level when it comes to the lamprey. So now the last thing, the subcellular models within HPP.
Just to say, Jeanette, you have a couple. Don't rush. You have a couple of minutes just to give you a correct on timing, because there's lots of questions from our audience.
Okay.
So no worries.
At least at this level, we use modeling to investigate mechanisms important for learning through synaptic plasticity. And when it comes to the basal ganglia system, where you map cortical activity to the behavioral outcome, then we have plasticity in this corticostriatal synapse, and this has been studied a lot. So if this is a striatal neuron with the dendrite, you have small protrusions here, and those are dendritic spines, and in these spines we have activation of biochemical signaling pathways because we have neuromodulation or we have activation from cortex, and these are the things that can be modeled. And this is just one example. We all know that in reward learning, the action has to come before the reward, otherwise you don't learn. And that's of course make functional sense. But some years ago, it was shown that this was implemented at the subcellular level, and even a dopamine signaling pathway and a calcium signaling pathway was sort of identified. And then we wanted to use modeling to identify how interactions between these biochemical pathways could support these timing-dependent reward learning. And to make a long story short, when we first simulated exactly the pathways that were suggested, we saw that they have to come sort of close in time, the reward and the action command have to come close in time, but there was no really asymmetry. It worked equally well if the reward came just before. So then we went back to the literature and to transcriptomic databases to look which other proteins are providing an interaction between these pathways and which are very much expressed in this kind of synapses. And then an example of that is this molecule called ARP, and when we put that into the model, we get this very robust timing sensitivity. I think this is just an example, but I hope it's an example that that shows that modeling can be used to identify targets for the subcellular level that is involved in synaptic plasticity. And as you might know, synaptic plasticity is actually something that goes wrong in all neurodegenerative diseases and most of the neuropsychiatric disorders. And when plasticity can't compensate, that's when you get symptoms. So to really understand and be able to interact and counteract plasticity impairment is key. So I just wanted to acknowledge that a lot of people have been involved in HPP work over the years, and of course also many others outside HPP have collaborated, and funding is also from various sources. So thanks.
Thank you. And thank you, because when I first looked at the end, as I said at that slide, I was like, yes, lots of women, which is just because it is so rare for me to see that in any team in general, in any sector. But sorry to give you those time signals, but I can see there's such activity from our audience. I have a really couple of short questions for the three of you together, and then I'm going to literally go straight into that fast track Q&A. So if I can invite back, please, your colleagues. Absolutely fascinating. Also, thank you, Jeanette, for all of that. And I know that was a lot of pressure to be so clear in such a short time, but you were very fantastically interesting. I suppose the quick question, and please for short responses, is anything you wanted to add? Now we've heard from all of you together, and you know, we say how interesting it is in the brain regions, their dependency, how they collaborate. Anything we just wanted to unpack a little bit more on that before I pass to audience questions?
Yeah, I mean, I think this thing how the different brain regions collaborate. I mean, when we build these functional models, we assume that cerebellum is doing this thing and basal ganglia is doing this thing and hippocampus is doing this thing. But I mean, they do depend on each other. So an interesting thing here, hippocampus, for instance, output from hippocampus go into part of the stratum, and then it goes into frontal cortex that then goes to the to the basal ganglia, and the same is true for the cerebellum, that it talks to basal ganglia via thalamus and also cortex, and also functionally, I mean, you can, while basal ganglia is selecting like an object or where to watch, then cerebellum is making sure that you have a smooth movement to keep your eyes on the, or you can, if you grab an object, that you can grab it in the right way, and so on. And the same is true for hippocampus, that even though hippocampus is doing sort of a map of the world, if you turn off the light and so on, and then also the basal ganglia is important, because then you orient based on
yourself. Absolutely, it's a great example.
Right. So they are really dependent.
Okay, I'm not going to ask you guys to step in. What I am going to say is, particularly because, and then I will take these questions, so I know our team is getting ready, particularly because you mentioned there the importance of interoperability, Jeanette, and the importance of being able to study these models and how the different models fit together in their relationship. Is there anything particular in the eBrains and the HBP project that promotes this, Michele, Eduardo? Anything that supports particularly that aspect of the work?
Yes, there is. As a matter of fact, eBrains is unique in allowing users to use different technologies to do multi-level simulations of brain regions transparently and very easily, without knowing anything about the coding or the actual neurobiology, in such a way that even a student can learn from using eBrains, can learn how we do a model, why we do a model, and how we can use a model. I think that this is the only research platform infrastructure that allows this kind of interaction.
Okay, thank you for that. Eduardo, just to be fair and balanced, anything you wanted to add on either of those aspects before I move to audience questions?
Yeah, well, I would agree that, there are so many tools in the eBrain that in the old times, before when we were jumping into these kind of models, we had to build all the tools ourselves, handcrafted. So the barrier was very, very high. And also it was very difficult to reproduce the work by other researchers. So in that sense, this is getting easier and easier with these kind of tools. And the eBrain is playing a role there.
Thank you for being specific there. So are you ready for intellectual bingo, ladies and gentlemen? Okay, so we are going to take some questions now. You need to be beautifully as concise as you can be. I don't know what's coming first, but we will be able to see them. There we are. You mentioned students there. That was something you mentioned, Michele. So there you go, from Ursula de Souza. I'm a grad student of computational neuroscience. Here we go. Where in the world are the emerging markets?
Well, these are actually two questions. Where are the emerging market, and who is doing the same thing that HBV is doing? I answered the last question. HBV is doing a very unique work in terms of merging experiments and simulation into a single platform. There are many other brain initiatives in the world, in the United States, in Japan, in China, in Canada, but none of them, and of course also the Allen Institute, but none of them provides the tools and services that eBrains is providing and will be providing very soon for users. So where the market is? The market is in clinical, pharmacological, and robotics applications. I think that those markets will benefit by a lot using brain simulation science.
Okay. I would say just raise your hand if we absolutely burningly need to add something. Otherwise, I will move on to the next question if we feel it's been amply addressed. Petru, Bogdan, Michele, how long does it take to simulate 1 second of hippocampal activity on a high-performance computer?
Good question. So, of course, it depends on how many processors you use at the same time. For our model of a rat hippocampus, it takes about 10,000 processors for a couple of hours. So it is a huge time in terms of absolute computing time, and this is why we need to reduce it the complexity of the model after we have built this, because this is our reference directly in connection with the experimental and the real thing. But we need absolutely to reduce the complexity in such a way that everything can go into a chip and doing their operation, higher brain function in real time.
Okay, thank you very much. I love that. That does feel like that game show with brilliance, doesn't it, section? So there we go. Eduardo, please, do you see that?
Oh yeah. Well, the question is about different learning mechanisms with different dynamics. So I would answer that the good thing with the simulations is that we have full control to all the parameters. So we can in fact do different simulations with different dynamics on the different learning rates. And we can explore how they complement each other depending on the parameters you choose. So in that sense, it gives you the freedom or the power to simulate all these things. So, but it's a complex system. So I would say that it is not only these learning rules. There are many other plastic mechanisms. So it's really quite a challenge to study all of them.
Okay, we've got a very active member of the audience there. There we go, Michele. And we do have questions for you, Jeanette. We're just giving you a breather, but they are coming, as you know. Would someone have to insert new variables in pharmacological simulation?
Okay, in principle, no, because what we need to do to model one individual or another is to change the values of the variables that are already in the model. We see this every time. I mean, there is not a single experiment in neurobiology that allow you to record from even the same neuron at two different instant of times and getting exactly the same response. So there is a normal physiological variability that we can take into account into the model. We don't need to add the new variables. We just need to adapt the existing variables values to represent a specific individual.
Okay, thank you for that. Let's see, there we go. Here we go. Jeanette, you see there the question. What are some behaviors?
There are some behaviors that simplified neurons can't reproduce in basal ganglia models. Yeah, I mean, non-linearities in the dendrites, such as what is called dendritic spikes, they can do really this non-linear integration of inputs. And actually, we are currently, as we speak, trying to make these neurons learn the so-called XOR problem, or this feature binding problem. And it seems that they can indeed do that if they are non-linear enough. But of course, a point neuron can't do that. So everything that relates that has to do with that dendrites are non-linear. This is difficult to reproduce with an ordinary point neuron. But of course, then you can just have several layers of point neurons, so you can fix it. But I think what is important with these different type of models is to actually understand what you lose at which scale. So I mean, this is important.
Right. Okay, so we're still good. We've got about 7 or 8 minutes of time. So there we go. It's great to have one that is good for all of you. So take a moment, and I don't mind who would like to.
Well, I will try to give my word on this. Well, of course, anything that we don't include in the model, it is going to be a problem sooner or later if we want to implement a whole brain simulation. So it depends what kind of resources we have to implement a proprioception rather, for example, language. Language, for me, it has a much higher priority with respect to proprioception, but it is an important issue. And I don't think that we have a good model for language. And when I say good model, I mean in the neurobiological sense. There are translators, Google translators, or any other kind of closed captioning for the TVs that do a very nice job in making translation or understand word or even talk, okay? But those are very artificial algorithms. They are not based on any actual biological circuit. And I think that it will be a great step in the understanding of how the brain works if we can figure out a network based on biological items, biological components, that is able to produce language. Proprioception is important, but I don't think it is a first priority at this point.
Eduardo, you want to?
Eduardo maybe, of course, very different.
But I would say that we are making an effort and doing step forwards to do behaving simulations. So in which we have the neural systems integrated with a behaving body. In that sense, that helps us to understand very well better how different functions are based on this proprioception and different mechanisms. So it's not only the neural system; it's also how we integrate it with the body. So I think there are now tools that allow to do this kind of things, but there is a lot to develop. So we need to develop a lot of tools, a lot of things to be there. We are not there yet.
Jeanette, anything to add?
No, not really. I was just thinking about some old study we did in the lamprey where it turned out that this sensory stimuli made it in the spinal cord, made it swim through a streaming water, but without it, it couldn't. So I think this is a great, I mean, of course, the surrounding is giving a lot of information that is used by the nervous system. So that's, of course, obvious.
Okay, thank you. Here we go.
Okay, deep brain stimulation. So actually, as I understand it, I think that many times, actually, one is using deep brain stimulation in Parkinson's in the subthalamic nucleus, STN. I think that is one of the most common targets. But I think it's very interesting because I think it's not really well understood why it works, but it works. Does it increase the activity where you stimulate, or it decrease it, or do you introduce some plasticity or something? But I think this is something that can also be explored with more detailed modeling of the whole system. And also, I think it would be very interesting. I mean, this deep brain stimulation has started to be applied to various neuropsychiatric disorders. And it's very interesting, especially maybe to manipulate also neuromodulatory systems, which are very influential in a lot of diseases. So I mean, definitely, yeah, in the basal ganglia, definitely STN and some other nuclei. But yeah.
I think if we have them, we've got time for 2-3 more questions, certainly. There we go. So we've got some more students. As you see, we started with a question from a student. There we go. Can you suggest me? I think you can help with that one, I hope. So who can suggest a learning path?
Well, I can start and saying that first you need to have a good follow a good course on coding if you want to be a professional in the field. Coding, computational coding means Python, the programming languages. Dealing with a computer is of paramount importance, of course, for simple computational neuroscience, and also with a good knowledge of the neurobiology, that the biophysics that is behind the biological mechanisms that drive how the brain work.
Yeah. I would add that, well, at the Human Brain Project, they are being produced specific courses for different topics related to the computational neuroscience, but there are also other summer courses around with a lot of online material. So I would say that the barrier is getting lower and lower. So before, it used to be that you would need to go to a specific lab to really acquire the skills, while now there are many resources around. So I think, yeah, at the Human Brain Project, there are specific courses, but there are others also, summer courses, which I find quite interesting to follow.
Okay. Okay, that's the last one. Fantastic. And then I will ask a last question to you all before I try and close this on time. So there we go, Eduardo.
Well, yes, how to replace the Baxter model with a soft compliant robot. Yeah, well, this is quite a challenge. I quite like the Baxter because I think it reached the industry. So before a control scheme was there. So in that sense, it is a robot with elastic components, but the accuracy you get is not as good as you would get with an industrial robot. And this is because you are people is using usually control schemes the same as with other industrial robots, and you should use different control schemes, more biologically inspired probably. when using these soft robots, well, this is a challenge. We don't even have models, I guess. So I would say yes. I would say that it would be really interesting. But we also need to be aware of how much technical work is behind any of these setups. So there is quite a long time, years of technical engineers setting it up. So it's, I would say, not easy to really go from one robot and apply that for another robot at all.
Let me just, I don't think anyone's going to be upset if we're 1 or 2 minutes longer. So I'm just going to give the team backstage time. If there's a burning question that you're going to think somebody out there is going to be frustrated not to have it answered. There you go. Good. Thank you. Good. Very good question. For sure. And we needed this one. So happy to hear your responses to that from Phoebe Gilbert. What sorts of ethical frameworks or paradigms?
Well,
even?
I mean, speaking from a computational neuroscience point of view, luckily enough, the ethical problems are very much reduced in terms of the usual problem, for example, in comparison with the usual problem that the experimentalists have. But there could be ethical misuse of the models. And what I can say is that we just follow all the regulation. In the Human Brain Project, there is a very strong ethical team that supervise and check all the work that we are doing within the project, and if it can have ethical issues. So far, no, but we are following their guidelines.
Okay. I'm going to park it there because I'm just going to ask you sort of an obvious last question before I close this for our audience. Do you have to pinpoint some future challenges concerning brain modeling and simulations? I think that would be very useful just to flag up a few of them as you see them. Jeanette, please feel free to begin.
Yeah, no, I mean, my thought is in that case to sort of repeat what I said, that it's both an opportunity, but if we don't grab it, it will be a big challenge to really make all these models interoperable so we can plug and play with them and in that way really build larger systems and explore different functions in different modules. So I think this is a big opportunity and yeah, but it's also a challenge because it's not so easy to go beyond your own little speciality, but I think we have to do that. And unfortunately, eBrains is supporting these kind of efforts.
Okay, thank you. Eduardo, what about you? Where do you see, what do you think about when you put your head on the pillow at night? This is the challenge. This is what...
Well, I would say brain and understanding how the brain works is in fact one of the challenges of this century. So, and all these tools are helping us to do that. Then when we have all these models running on supercomputers, I see another challenge in taking this huge model and put it into a domestic robot at home. So there is another challenge of really taking it to the market and using it for domestic things, helping us. Mm-hmm.
Michele.
Yeah, so I mean, those two issues presented by Jeanette and Eduardo are very important, extremely important. What I see as a real challenge, additional challenges for me, is that we actually need to go to humans, and then we want to model a brain of a human. In this case, what I'm extremely eager to wait and get my hands on is data from humans, because it is the only way in which we can say our model is a relatively good representation of how our brain works. So far, we have been working with the rodents, but mouse and rats are not humans. I mean, even if they are 99% genetic similar to humans, we still have something different. And I think that once we have the data, we can really say we have modeled the human brain.
Thank you very much. And interestingly, didn't we, when we all met before and we were, working together for this webinar, we were saying, should it be 75 minutes? Should it be 90? And I can see that we could have made it 90, couldn't we? Because there is an awful lot of interest, partly just because you were so splendid in your presentations. And I really do thank you all very much. And I know it was quite a challenge to really condense things into the time that we had. So, ladies and gentlemen, I will thank these very good speakers, Jeanette, Eduardo, Michele, on your behalf. And of course, I also thank all of our lovely audience for their brilliant questions and for being with us for our monthly series. And thank you also to the comms and the digital teams behind the scenes who work very, very hard to synthesize the content and to synthesize the questions that come in. Of course, if you've enjoyed the discussion today, then just keep following us. There are more editions. Now, the focus that's coming up in the next few editions will be on the work sort of from the last few years, but also in effect what the project is trying to achieve in its final phase. So do keep your eyes on the website if you want to know what's coming up next and when. Of course, as I said, if you've been inspired by today, do let your thoughts fly free via Twitter, #BrainMatters, and of course, at any point, just reach out to this thriving community. That is really important for the project. So that's it from me, Katrina Sickle, to all of you. It's an absolute pleasure to share the screen, the virtual space with you, Jeanette, Eduardo, Michele, and with our lovely audience. And I think we can all, in our regal or happy fashion, thank everybody and wish them all a very good evening. Thank you very much. Until the next time.